And good evening to the participants and trainer. So today I would like to welcome you all to the 20th day of the 21 days long training program on biological data analysis and bioinformatics. So today the session will be taken by Dr. Rupsanatan Mandal, who is currently working as an assistant professor of Genetics and Plant Breeding Department at Uttar Krishi Vishwavidyalaya, and also a visiting scientist at CIMMYT, Mexico. So, sir has already taken some very informative sessions on bioinformatics. So, yesterday we got a query about a session on gene annotation and sequence alignment. So I would like to mention that the session has been already taken in first and second class by Dr. Rupsanatan Mandal. So I would like to also mention to the participant that please don't be worry. Actually, we will provide all the recordings. If you miss the classes, you will get the recordings. Furthermore, if there is any confusion, I would like to request Rupsanatan sir kindly clarify it to the participant. So thank you all, and I would like to request Rupsanatan sir to kindly start the session. Thank you.
Thank you. Good evening, everyone. So today this session is very informative and it is very important. We are working on the genome-wide association or genome-wide study. Okay. In the first two class, I have already discussed how to download a gene from the phytosome. But today I will show how to download the whole genome sequence, how to download the individual protein file, how to download the individual sequence file when the sequence are in batch or a group. So previously we have learned about the single gene, but today we will download from uh, different number of gene from a single gene. We know that different types of derivatives. They have the homologous gene and they have the orthologous gene. So when we see the database, we will found there is lots of gene are available. Those are showing the similarity. They are showing 90%. They are show the 100% similarity. Okay, but which gene we can download and whatever the importance of the different gene in those are stored in the database. So today we will learn how to download these gene sequences from the phytosome and what is the gene annotation. I heard one message from in group. They mentioned the gene annotation, gene alignment. I don't know what she want to ask. Alignment means when you have one sequence, you just blast it, and that blast can align the sequences automatically, and they give you result on the base of alignment. If he or she is talking about the multiple sequence alignment, that is different thing. But gene alignment is only the if you have the gene sequence, you just blast it, and that is called the alignment through the any portal. We are using NCBI blast, but there are different lots of blast. You can also phytosome for blasting. This is also called alignment, and then alignment give you the different types of result. On the basis of that alignment, you can found your own gene. Okay, and gene annotation means gene annotation. There is different types of uh, method involving. One thing, you have your sequence, you can blast it, you identified that what is your gene on the basis of sequence alignment. So which one is show that 90% 99% similarity, that's mean your gene is that. This way you can convert your sequence, genomic sequence, into protein, then you can align. That also gives you the good result. If you want to identify the function of the unknown gene, then the process is different. Okay, and so gene annotation is one or different types of process involved. What you want in the gene annotation, I don't know. If you have the sequence, you can just simply blast it. That's mean it's annotation. If it's you need more details, please follow today class. Okay, I will learn a step by step. Okay, how you can download or how you can found that different types of gene mechanism. Okay, so let's start. I'll share my computer screen. One thing, I'll show you step by step. Follow this. If you have any difficulty, and another thing, participant, please take a notebook and write every steps. I know that most of the people are not following me in computer. Okay, they are just listening me or watching me. So when they are doing second time, it sure it will you will face some problem. So that's why when I am talking about different things, you just write on your notebook. That will help you in future. Okay, so previously we have discussed about the phytosome, but I also show you different how you can download the bulk gene sequences from the phytosome sites. Okay, for that, first we go to the phytosome site. Okay, now another one thing, from that phytosome site, yeah. So this is the interference of phytosome site. Before going to the phytosome sites, what you want, you need that. So today, for one example, I will just download HSP70 gene from the tomato. Okay, so and now we can from that gene we identified which are the duplicate gene using the phylogenetic tree. How we can make that phylogenetic tree, we show the different group of gene. Okay, so like that. And previously I have already discussed about the mega software. Also I have discussed about the itol. So everything I will use today. So first we need to download the gene HSP70 gene from the tomato genome. Before that, I know that you need to go NCBI site homepage. So this is the NCBI homepage. Here you can select the gene. Here write your gene name HSP70 gene in tomato. So here this is, you can see this is the hits of protein 70. and there is the different lots of protein of HSP gene. Okay, so you can click on either here, this is the reference sequence, or click here. Okay, so I'm just click on the reference sequence. If reference sequence are not available, then you click on the below sequences. So there is the gene annotation. You can see whatever the gene annotation result in through the NCBI. Okay, so where the gene is present, you can see this is the HHP gene, where it will be the present. Everything is given here. Okay, and through the NCBI you can download that image from here. Okay, FASTA sequence, PDF, SVG, everything. So I'm not bother about that. I'm just scroll down below, and here you can see the mRNA and protein. That protein or mRNA sequence is there. So either one we can, this is the protein sequence or mRNA sequence, start from the this and end this one. So either you click just one of them. I'm just click here. Now you can see there is the one page that is called the protein information page. Okay, and now you go to the first tab. So you can see this is the FASTA sequence of the protein. Okay, so you don't need to save this protein. You just copy this protein sequence from here. Okay, now go to the phytosome. After going to the phytosome, here you can see I am already logging. Hi Rupsanatan Mandal, it is written here. So when you are first time using this phytosome, you need to create an account which is freely. Just click on the right side. There is one is logging option. Click on that logging option. You can found create new profile and then register yourself with your email ID. Then you can logging into the phytosome. After then you blast it. If you need the large number of sequence download together. If you don't want, if just single by single, then you don't need to log in. But if you download the lots of sequence from the single click, then you need to log in. So it's okay. This is the interference. Now you go to the tools and blast option. Okay. Now we want to search the tomato genome. So you type the tomato. You can find there is the different version of sequence available. Which one the latest one? You click on that. Solanum lycopersicum ITA G 5.0 is the latest. Okay, so I'm click on that. Now I'm paste here the protein sequences and go. So you can see lots of gene are present. Okay, there is the, but today we identified, or today we download or take 10 number of genes. So first 10 number gene we are selected. Now what is the basis of that selection? So remember it, the cutoff value is the 1 into 10 to the power minus 4. 1 into 10 to the power minus 4. E value 1 into 10 to the power minus 4. So you can see these all are up to that. This is up to that. This is the 1.6 into 10 to the power minus 7. So this actually above the cutoff value, and below that, that is 4.61 into 10 to the power minus 4. So that is actually above value of the 1 into 10 to the power minus 4. So that's why below that we are not selected. Up to 1.60 into 10 to the power 6 we are selected. So these are the cutoff value. So you can just check this box which number you want to select. Okay. So then you see that one option will be visible, that is add to cart. Click on that, add to cart. So 26 numbers of sequences actually add to cart. Okay. Now you can download all the sequence together. Okay. So if I am doing all the 26 sequence here, that will take lots of time. Okay. For not downloading, collection some information, collection some different types of making file, it will take lots of time. That's why I am selecting only 10 sequences. Okay, so I'll take only first 10 sequence. I am just showing you how you can select the based on the cutoff value. So when you want to genome-wide study, you need to check the cutoff value above the 1 into 10 to the power minus 4. Okay. So now I'm deleting all that sequences and I'm just select only 10 sequence, first 10, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Add to cart. Then go to your cart, then select all 10 and then click on download. Okay. So what I need from the download, I need the genes. So click on genes and go option. Now you can click on the attributes. You can see this type of picture is available. Here click on attributes. You can see what you want to download. So first we want to download the sequences of peptide sequences. Okay. Last time we have download individual sequences. Here we can download 10 sequences together. Okay. So just click on that peptide and here go to the result section. Click on go. So that already all the protein sequences is download here. Okay. So now next I'm just go to the attributes and then I need to the whole gene sequence. Here I am just peptide sequence. So whole gene sequence means unspliced gene. Okay. Whole gene means unspliced gene. You click on the unspliced gene, go to the result section, click on go. So on all unspliced gene are downloaded. Now click on the again attributes. If you need to see DNA sequences for gene structure, you know that gene structure you need the cDNA sequences and whole gene sequences. So here you just click on cDNA sequences, then go to the result, automatically downloaded all the sequences. Okay, it's Very simple. Okay, this way you can download it. But now we need to some different types of information regarding these genes. Okay, that information we can pick through the one by one. Okay, so first see the download file. So this is our protein sequences of 10 genes. This is whole gene sequences of 10 genes, and this is the cDNA sequences of whole gene. So these three files we need. Cut it. Paste here. Okay. Now see the first sequence, that is the peptide. So rewrite this sequence. Peptides. Peptides for HSP 70. Now this is the cDNA sequences, and this is the gene sequences. So gene sequences and this is called the peptide sequences. Sorry, cDNA sequences. cDNA sequences. Now we need to change. We want to change the name. This name, we want to rename these sequences. And first we need to change the, you can see in peptide sequences, there is asterisk sign. That is the stop codon. So for further analysis, you need to remove this asterisk sign. So open this page, select Ctrl H, write here asterisk and replace with none, nothing. So you can replace all, and you can see that all asterisk sign already removed. So after that, we need to convert these sequences. These are all sequences in FASTA format. So we want to change the name of these sequences. So if you want to change this name of the sequences, you need to change this file FASTA to table. Okay. So if you want to change this table FASTA to table format, then you need one software that is called the TB tools. That TB tools freely available, one of the Java based software. Remember one thing, if your computer is not supported the Java, then you don't you have not run this software. Okay, if your computer is support the Java, then you can run. And another thing, your computer must be a good RAM and good gigahertz. So then you can run this software. Otherwise, it will not work very good way. Okay. So in TB tools, there sequence FASTA sequence toolkit and choose that FASTA tools. Okay. So where you want to just, you can see here different types of option available. Okay. So extract FASTA, extract basic, extract subsequence, FASTA start. Okay. Simply FASTA ID. Here too you can see FASTA to table convert. So click on FASTA to table convert. Now here you can see the input file. So our input file is peptides. Drag here, close it. Now we need to FASTA to table and we want to see here. So convert this. Okay. So you can see your gene is converted. You can copy and now open one Excel file, paste here. You can see this is your sequences and this is your, this is gene ID and this is sequences. That sequence peptides. Sorry. Okay. Now, if you want to rename gene ID, you can want to rename this gene ID. So you can rename and now you can give that name HSP70 for Tom underscore HSP70 underscore 01. So this is your sequences. Okay. So now you need to reconvert into the FASTA sequence. You have already renamed these sequences. So you just copy these two sequences, go to the TB tools, FASTA. Now FASTA to table convert or table to FASTA convert. Now here you table to choose table to FASTA. Now sequence input, paste here. Okay. And output, convert it. So here you can see your table to FASTA, it is already converted. And paste here. You can see your all sequence ID name will be changed in the rename. Okay. So save it and rename this sequence. New PEP ID. Okay. So this is the new PEP ID. Okay. That's all. Now we want to one phylogenetic tree, and that phylogenetic tree will show you whether there is any duplicate gene or not present or not. On the basis of that gene, we can prepare, or we can show our gene visualization. Okay, so for that we all know that we need to the mega software. Okay, so this is our new peptide sequence. Now we are opening mega software. Okay, just click on the mega software, then align, create or edit new align. Now create a new align, click on OK. Now from protein, then import the protein from the sequence file, where our sequence is the gene visualization new peptide ID. You can see, delete the last one. So this is the sequence, alignment sequence. Okay, now you need to save this sequence, so export the alignment as a mega format. Okay, so your destination file, that is the gene visualization, change folder, right here mega, we name this HSP70. So HSP70. Okay, so it will save. Now you can close this file. Now we need one to phylogenetic tree. So construct the phylogenetic tree. So choose your original file. Now I'm using here 10 number of bootstrap or replication, but you need to at least 1,000 times replication. It will take time. That's why I'm showing taking 10 numbers of bootstrapping. So it will, that phylogenetic tree will be like this. Click on the OK, and it will be creating the original phylogenetic tree using the bootstrapping method. Okay, so your phylogenetic tree is already created, but we are not taking this phylogenetic tree. Okay, we need good one, so that's why I am just save this, export this tree file as a quick file. Okay, including the branch length and bootstrap value. So okay, you can found this. So save this one in the gene visualization folder. So this is the tree. Okay, save it. It will save. Now we open the ITOL folder, ITOL tool. So upload my tree, choose the file. It will in gene visualization, Megha. This is the tree file. Okay. Now upload it. Okay. So ignore the branch length. Where the ignore the branch length. Okay. So this is your uh, phylogenetic tree. Now you can see you can see this is this phylogenetic tree. You can see there is the one single group, this one the single group, there was the one, and this one the one group. So this one, two, three, four group. Okay. But in the four group, there this is the one of the major group. Here one of the single group, this is the single group, this is the another group, and this group also divided into the two this subgroup. Okay. Now we need to color this. Okay. So label the branch color. I need choose also the label this color also green, choose it. Now second, this is the branch color. I need to green, choose green, and so label green. Now this group, this branch color will be red, choose red. Okay. And you can select this also label. So this way you can label all the So you can see this is our phylogenetic tree. Now we save it in circular manner also. This is circular. If you want, this is rectangular. This is the unrooted. So I am want to rectangular. Now I want to export it. So click on export, and I want it is a PNG file with 600 dpi. So export it from mega to ITOL. You just get. So it will be, you can see this is your phylogenetic tree. Now here you can, from this phylogenetic tree, you can found there is the three group of proteins. Okay, within the HSP70 there is the three group. One is blue color, another one is the green color, and third one is the red color. Okay, now within the red color you can found some duplicate gene like this. Please see this node. So this HSP 10 and 9 are duplicate, HSP 74 and 1 are duplicate, and HSP 11 and HSP 8 are duplicate, and HSP 6 and HSP 3 are duplicate. So these are the duplicate gene. Okay, so you need to write in your you need to write this gene ID in your notebook for further when we want to visualize the gene on chromosome. Okay, so that's up to this. We are finding the duplicate. Those are the duplicate. Remember this. This is the same gene. Okay, we are first we are downloaded the HSP70 protein, but that protein have the some homologous gene. Okay, maybe some orthologous gene also there. Orthologous and homologous we can decide it through the different way, but today we are not discussing. But here we are on the basis of phylogenetic tree, we are found these gene may be duplicates. Okay, so up to this these genes are duplicate up to this. Okay, now we have already saved this phylogenetic tree. Now we need to visualize this gene. Some information are required. So for that information, you need what? One Excel file. Okay, and that phytosome site. For that phytosome site, you need to go your home page. Okay, this is your home page. Okay, now you need to click on gene view report. Okay, from that, when you are opening that gene sequences, here you can found some information. That is first one thing you can see, this information you need. So why this information is need, I am showing you. Open an Excel file. Okay, so I have already one Excel, that is the sequence. So I'm just copy these two column. paste here. Okay. So that first gene, you open your phytosome. You can see 11, then it's showing the colon. Then you found the gene start point and it is the end point. And it is always presented in forward and reverse. But for the gene position, we need these three information. So copy it or copy from, sorry. Okay. Go to your Excel, paste here. And you can see this gene ID is 4.155.4.1. No, this is the 0951. So right here. Okay. Then open another gene report. Go to the genomic section. Here you can see so this is the 554. So 554. Okay. Then go to the phytosome. This way you need to collect all the gene information. Without that information, you cannot proceed. Close it. Open the next. 2, 3. The ID is 3830. 3830. Paste here. To close it. Then 4 number. 3820. 3823. 4 number. Then you need to 5 number. 5. Now 6 number. Seven number. Seven number. Now 1, 2, 3, 4, 5, 6, 7, 8 number. Eight, 4, 3, 5, 8, 7. 9 number. And 10 number. This is the 10 number. Now here we found that 11 number of gene. So we just delete this 6 number. Okay. So now it is 11 number. Okay. But our information required chromosome number CHR. First gene is present in the chromosome number 1. See here. This is the information chromosome chromosome number. So first gene is present on the chromosome number 1, second also 1, third also 1, fourth also 1, fifth one is 3, 8, and that one also 8, then it is 11, then it is 4, then it is 7. So you can see same gene, they are distributed on the different chromosome of the same crop. Okay, so this is the genome wide distribution. Now the start point where the gene actually start and end. So start is, sorry, cut here, cut here, cut here, cut here, So this is the chromosome number, this is the start point, and these are the gene ID. Okay. So our information is the gene ID, chromosome number, start point, end point. So this file will be saved. You can copy this and paste in the text file. Text file, and you just save it. Okay. Why this is the problem? Okay. So rename this folder gene position. Now you need the another information, color of the gene. Okay. So copy this. This is the gene name, and this is the RGB color, RGB. Okay. Now see, before that RGB color, you need to see your phylogenetic tree. Okay. So that phylogenetic tree, what you have given the color green, blue, and red. So you can see the HSP5 is the blue color. So here HSP5. So for HSP5, you need blue color. Go to the cell, more color, custom information. Okay. So if click on blue, blue. So you can see the RGB is 5847,199. So here you can 48 5847,199. 5847,199. Then close it. Go to phylogenetic tree 07. That is the green. So custom in green RGB is 24, 232, 54. And then others is the red. So red RGB is 237, 31, 80. Now 7 is blue. Blue is 58, 47, 199. Okay, 7 is the green, 24, 232, 54. Now our remaining all are red. 37, 31, 80. So just copy and paste here. Okay. So this is your another folder. You just copy. This is known as the color of gene. Color of gene. Paste. Now another file is required, that is the length of the chromosome, full length of the chromosome. So full length of the chromosome you can get any literature or any database of the NCBI. So you need to find this full length of chromosome from that literature or database. I have already collected, so I am just copy that folder and here paste it. Now I want to show that what is the chromosome folder. That folder is this is the chromosome name. In tomato there are 12 chromosome, and this is the chromosome length of this one, the chromosome number, 12 chromosome number, and this is the individual length of that chromosome, and that chromosome length approximately in base pair. Okay, this is given in the base pair. Okay, so this file, till these file are completed, now we go to the TV tools. In graphics there is show gene on chromosome, then gene visualization advanced. Okay, so there is the different required different type of folder. First, drag your length of the chromosome. So this file I am just drag here. Okay, then gene position. So gene position file I have already prepared. Okay, then the link gene. Now I am not giving the link gene information. Then I am giving the color of the gene. Okay, so color of the gene file. Okay, close. Now if everything is okay, then start. So it's showing some error. Please see. So, okay. Color. Maybe in position of the file there is some problem. So I save as Subhradip. Okay. Position. Yes. So you can get your chromosome mapping. So you can see this is the 12 number of chromosome 1, 2, 3, 4, 5, 6. Here you can found your gene. Okay, this is the gene number 10. This is the gene number 11. This is the gene number 9. And others gene you cannot found because I have collected the chromosome length from one literature, and that literature is not true. So that's why you can download your chromosome full chromosome length from the genome. So in phytosome, go to the genome, okay, and then you type your desirable crop. Here it is Solanum lycopersicum 5. Click on that. Here download, go to download option. Okay, proceed. Here you can see lycopersicum annotation full in text file. Okay, and FG file lycopersicum CDS FGH file. That FGH file. Okay, and this is the whole genome information. Data release lycopersicum full. You can download this file, this FGH file, or this data release policy file. Okay, that is the, and this is the assembly file. So you can download this assembly file also. Add to cart, go to that cart and click on download. Browse download. It will take some time. So after download, you just click on that folder. You can get chromosome wise sequences. So from that sequences, you just get your exact chromosome location. Okay, and here you can choose the chromosome bar color as your preference, like so this is your chromosome. Here you can change your front gene front level. Okay, so in front size it will be like that old, so font size will be increased. Okay, so this way you can design your own gene. Okay, or gene visualization on chromosome. So if you have the gene and you have the information where gene is start, where the gene is end, and then you put this information and you can get such type of the visualization using the TV tools. Okay, so if you have the one chromosome, you can also putting the all genes in one chromosome. Okay, so this way you can design this type of map. You can found those are QTL mapping like that, but without QTL mapping, only the gene information is your. So if you are sequencing one gene, then you can uh, make the phylogenetic tree with the other similar type of gene, and then you can design your own visualization of the gene this way. So I know this session is quite difficult to remember all the step by step. I think organizer have the recording of this session. You can record the session and you practice at your home. If step by step, it will definitely give you the result. Okay, so if you have any question regarding this practical session, tell me. So I think no one any question. So if you have any question regarding this session, also ask on WhatsApp group. So thank you all of you and good night.
Thank you, sir, for this very informative session, and I hope that will be beneficial. So now we are closing this session, and tomorrow it will be the last session of our training program. So I would like to request all participants to be present tomorrow, and also I would like to request Rupsanatan sir kindly to the last session. Okay, thank you all.